Hello friends, welcome back to my YouTube channel Nestin Padasale. So in this video, I'm going to provide you more scoring topic or highly important topic with regarding to a GATE 2024 biotechnology examination. If you didn't even start a preparation, you can study or you can prepare notes only for the topic that I'm going to discuss in this particular video. So without any delay, let's get into the video. First is vector. You need to know about the application. At the same time, you need to study about the size. Match the following type of questions can be asked from vectors. Next is plant hormone, both the precursor as well as the function. Again, match the following type of questions will be asked from plant growth hormone. Next is amino acid, starting from whether it is positively charged or negatively charged or polar, non-polar. And also you need to study about the biosynthesis of amino acid, few significance about amino acid. And also you need to study about which amino acid is having 2 chiral center, 3 chiral center, peptide charge calculation, and also which amino acid will be present interior of protein, which amino acid will be present on the surface. Okay. And also you need to study about the protein structure, primary, secondary, tertiary, quaternary. Okay, so while studying quaternary structure, you need to study in depth about the hemoglobin and myoglobin. Next is antibiotics. So antibiotics, you can expect match the following type of questions. And next is translation inhibitor. Both the prokaryotic and eukaryotic, you need to understand the translation inhibitor. So in Watson book, there is a tabulated column is available. So from that only questions are asking each and every year. One questions will be coming from translation inhibitor. Next is PCR. Basics of PCR, about Taq polymerase, the uniqueness about Taq polymerase, which activity is not present in Taq polymerase, and about thermostable, some other thermostable polymerases, and about few numericals that you need to know about PCR, and types of PCR. You need to study about the real-time PCR and reverse transcription PCR, very, very important. Next is pedigree analysis. So sometimes they will be asking about they will be giving you a disease name and they will be asking you to match the mode of inheritance, which is autosomal dominant, which is autosomal recessive, Y-linked inheritance, criss-cross inheritance, like that. Cell cycle and their regulations again and very important. So please don't skip while studying cell cycle. You need to study about the regulation process also. Next is genetically modified plants and animals. So recently GM acid was released in the environment. So just pay enough attention while studying genetically modified mustard. Which institute has made them, what is the use, and which gene has been incorporated. Next is gene transfer methods both in plants and animals. Okay, so plant tissue culture is also important, animal cell tissue culture is also important when it comes to gait, and plant biotech is also important, animal biotech also important. While studying gene transfer technique, you need to know about the agrobacterium mediated gene transfer. You need to study about the TI plasmid, their size, tDNA, and also about the mechanism and which gene and their function. Next is chromosomal banding technique like G banding, R banding, and what are the reagents that are employed. Next is mutation. While studying mutation, you need to know about the Ames test. Okay, and also some other thing like transversion, transition, and all the chemical base analog you need to study about. Next is chromosomal aberration. Both single gene genetic disorders, chromosomal aberration in the sense euploidy and aneuploidy. You need to study about the numerical as well as the structural. Both are important. So I think recently in GATE 2022 biotechnology, there was one question asked with regarding to the structural chromosomal aberration. Okay, so don't skip that particular topic. Chromosomal aberration is very, very important. Next is single gene genetic disorder. So in single gene genetic disorder, you need to study about triple nucleotide disorder. Okay, what is the triplet nucleotide that is repeated and whether it is present in coding region or non-coding region like a Huntington's syndrome. Next is chromosomal disorder like Down syndrome, Edwards syndrome, and some other chromosomal disorder are very important. Next, telomere and telomerase, both are important. What are the specific unique activity about telomerase, whether it is a DNA dependent or whether it is RNA dependent, whether it is having a reverse transcripted activity or not, like that. And telomere, you need to know about the structure, sequence of human telomere. In one time, the GATE examination and also in some other examination, they are asking about the sequence of telomere. 5' to 3', you need to know about the sequence. Next is again cholera toxin, their mechanism, whether it is G stimulatory or whether it is G inhibitory, like that you need to know. Next is cell signaling. Cell signaling is very important. You need to understand what are the secondary metabolite, secondary messenger, primary messenger, and about other cell signaling pathway. Don't go in depth about, at least try to figure out the tabulated column that is available in the Albert Cell book. Next is cytoskeleton, again very important cytoskeleton, like myosin, actin, microtubule. There were few drugs that will be inhibiting, that will be targeting microtubule, few will be targeting microfilament. You need to study about the drug also. Next is again formula, degree of reduction of ethanol, lactic acid, glucose. Okay. Next is antibodies. All the five antibodies like IgG, IgA, about the structure, which is a dimer, which is having a secretory component, and mostly functions related questions only will be coming. You need to study about the pepsin digestion, papain digestion. At the same time, you need to know about the gel electrophoresis. Like how many band will be getting when we run the agarose, when we run IgG or IgA in the gel, like the questions will be asking. So have an in-depth analysis about antibody. You can expect 2 questions from this particular antibody. Next is hypersensitivity reaction. 4 reaction are there. Only tabulator column alone study. I had to study tabulator column alone. And from the tabulator column only they are asking the question also. Next is autoimmunity, which is an systemic, which is an organ specific. Okay, you need to study about autoimmunity is again an important. Don't skip autoimmunity. Next is vaccine. Okay, study about all the vaccine and their example. And also you need to study about Covaxin and Covishield. Next is a complement system. 3 pathway of complement system. Complement system is again very important. Next is MHC. Class 1 and class 2 MHC, which will be presenting endogenous antigen, which will be presenting exogenous antigen. There is a pathway is also been there given in the QBA book. So kindly do study about the pathway. At least look on the diagram alone. That's well sufficient. So MHC is also very important. Okay. So if you can't able to cover up the entire syllabus, the entire thing that was given in the QB book, there is a tabulator column alone will be there. Just mug up or just study that tabulator column alone. Next is transplantation. Transplantation in the sense, so you need to know about isograph, syngraph, all those things. Next is allotype, isotype. These things are very important. Next is your paratope, epitope. Sometimes simple question also do ask in gate examination. So don't forget to revise the EC concept. Okay. The revision is very important, friends. So don't forget to revise at least twice. Even though if you are preparing only for 10 days, you need to split the 10 days into 9 days and 1 day. 9 days you need to prepare. At last 1 day before the examination, you need to revise the concept. Next is agarose gel electrophoresis. So while studying agarose gel electrophoresis, you need to study about there is a diagram is given which will be running fast. Concatemer, linked, supercoiled and nicked, which will be coming first. So they will be giving the gel itself and they will be marking A, B, C and they will be asking you to match them. Recent days, this is a trend I think so. So agarose gel electrophoresis, you need to study which will be coming first. Supercoiled, nicked, linear, concatemers. Okay. Next is STS page technique. Next is immune cells and the organelles, primary lymphoid organelle and secondary lymphoid organelle. Next is super antigen. Hybridoma technology is again a very important thing. Okay. Next is cancer, types of cancer and drugs for cancer and viruses that are associated with cancer is also important. Next is apoptosis and you need to know what is the difference between apoptosis and necrosis. And while studying apoptosis, also you need to know about extrinsic pathway and intrinsic pathway. Next is DNA polymerases, both in prokaryotes and eukaryotes, the type. Some you need to know about the abbreviation also, like DNAG, what is the role of DNAG, what is the role of DNA, like that also you need to know. Next is with regarding to RNA polymerase. RNA polymerase is also very important. So kindly do study about RNA polymerases also. Next is types of DNA. A form of DNA, B form of DNA, Z form of DNA and types of RNA. Like your mRNA, tRNA. tRNA you can get the match the following type of question. So while studying tRNA itself, you need to study about the selenocysteine loading, glutamine loading, pyrrolysine loading, all those things. Next, miRNA, siRNA, PV interacting RNA. Next, purine and pyrimidine. The structural components of purine and pyrimidine. Next is chromatography. At least try to cover about ion exchange chromatography and gas chromatography. Next is DNA fingerprinting, footprinting are very important. So try to know the principle and the procedure. Next is molecular marker, which is codominant, which is dominant. Next is viruses. So viruses is a very important topic. You need to know about the genome. First try to cover up entire concepts with regarding to COVID virus. Then go for other common virus. And sometimes they'll be asking you the virus and the route of entry and the virus and the specific host cell receptor. Okay. Next is gram positive bacteria, gram negative bacteria, difference between them. Next, prokaryotes, eukaryotes, archaea, the difference between three. Next is replication, transcription, translation. I know these three are bulk topic, but you can get at least one question from each. If you can't able to cover up the entire thing, study important thing only. Don't go for textbook. If you're having Pathfinder, that's well sufficient for GATE examination, I can tell you. Replication, you need to concentrate on initiation and termination. In transcription, you need to concentrate on RNA polymerases and initiation factors and their role. And also, you need to study about prokaryotic transcription and eukaryotic translation, the Shine-Dalton sequence, Kozak sequence, all those things. Translation, again you need to study about genetic code, tRNA and about the translation termination process. Okay. Next is disaccharide hydrolysis like maltose. What is made up of maltose is made of like cellobiose and hydrolysis, it will be giving what product, like that they will be asking. Secondary metabolite, both the precursor, the pathway and their application. So sometimes 2 or 3 questions will be asking from their application. So this drug is employed for treatment of which disease and it is taken from which part of the blood. You need to study about the thing. Okay. Sometimes one question has been asked like vincristine, we all know it is taken from Catharanda roses, but it is coming under which secondary metabolite, whether it is an alkaloid, whether it is a terpenoid, like the question has been asked one time. Next is microbe and their application, like which microbe is responsible for fermenting yogurt, which microbe is responsible for the industrial production of citric acid, which microorganism is responsible for producing penicillin. Next is herbicide and the target enzymes, again and very important. Next is peptide sequencing, like which cuts in N-terminal, which cuts in C-terminal, which cuts in trypsin, what is the role of trypsin, what is the role of chymotrypsin, carboxypeptidase, pepsin. Next is restriction enzyme digestion. Again, you can get one questions from restriction enzyme digestion problem. Next is blood grouping. A, B, O, blood group, H antigen. Next is recent in CSIR December 2023. One question is asked in 4 mark in blood group. Next is DNA repair mechanism and the diseases if that particular repair is not working correctly. Next is recombination process. Next is microscopy. SEM, TEM, confocal, phase contrast, like that. Biochemical pathway, you need to focus on location and the energetics alone, just concentrate. No need to go in depth. If you want, you can cover glycolysis, Krebs cycle, C3 cycle, C4 cycle, CAM cycle in respect to plant, you just cover. Next is protein sorting, again an important thing. Next is enzyme inhibitions, competitive, uncompetitive, like you need to know about the graph and what about Vmax, Km, and also you need to study about the example also. Next is electron transport chain. You need to know about 4 complex and the inhibitor. Next is post-transcriptional modification, post-translation modification. Both are highly important. Post-translation modification, at least try to cover about glycolization and phosphorylation. Because not all protein can be undergoing phosphorylation. Only some has the capacity to undergo phosphorylation. Same. O-link glycolization and N-link glycosylation are also important. Next is mRNA decay. Enzyme used in RNA tech, like exonuclease, endonuclease, alkaline phosphatase and protein kinases. Operon, lacoperon, tripoperon, arabinosaperon. Next is melting temperature, TM value. Your DNA denaturation, renaturation kinetics, if possible, do cover about histones, nucleosomes, also very important. Next is protein denaturation agent, Ramachandran plot, which amino acid will be coming under disallowed region, allowed region. The graph is there, the like graph will be there, 4 quadrants will be there, now that one. And about pi angle, psi angle, the glycine, proline. Okay. So NMR, X-ray, CD are important. Definitely I can assure you questions will be coming from which technique is used for protein three-dimensional structural analysis. It will be coming in the multiple select question. Okay. Next is period diseases and gene knockout technology. So while studying gene knockout technology, try to cover up Cre-Lox-P recombination. And yeah, these are the important topic that you need to study. And don't forget to study immunotechnics. At least cover ELISA, ELISpot, that's it. Okay, if you can't able to cover. BLAST, you need to cover about BLAST programs alone. BLAST N, P, T, BLAST N, like that alone, you just cover. These are the important topic. Okay, so please study all the topic that I had discussed in this particular video. All the best for exam.